on behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor, Gyros, I'd like to welcome you to Automating Pharmacokinetic Bioanalysis at Nanoliter Scale, Strategies for Development, Validation, and Transfer. At this time, I'd like to introduce our moderator for today's event. Carolina Oosterlund joined Gyros in 2003, working on MALDI mass spec applications and then immunoassays using Bioanalysis CDs. She has been providing technical support for the Gyrolab immunoassay platform in clinical and bioprocess development for 8 years and was instrumental in establishing the technology in North America and Europe. Her experience covers applications for pharmacokinetics, immunogenicity testing, biomarker monitoring, impurity testing, and product quantification for therapeutic protein development in both regulated and non-regulated environments. Carolina currently manages the Gyros European Field Application Scientists, as well as providing technical support to the efforts of Gyros Asia Pacific distributors. Carolina is involved in working groups such as the Global Bioanalysis Consortium and the 21st Century Bioanalytical Initiative. She earned her Master of Science in Chemical Engineering from Uppsala University in Sweden. And with that, Carolina, welcome. The ball is yours.
Thank you. I would like to start with welcoming everybody to this webinar, and thank you for participating. This webinar today will be focusing on PK bioanalysis, and we will cover strategies for development, validation, and assay transfer to CRO. We will see an example of the Universal Assay format used to support a preclinical study, as well as the specific assay format used to support a clinical assay. We have 2 different speakers from 2 different companies on the agenda. I will start with a brief introduction to the company Gyros AB and also to Gyros immunoassay platform. Then we'll have a presentation by Sufyan Makboul from Novimmune in the UK, and then a presentation from Emily Escoffier from Novimmune in Geneva. And after that, we'll have questions. But I will also address questions after each presentation, and please feel free to type in your questions throughout the presentations as well. The company Gyros was founded in 2000 as a spin-off from Amgen Pharmacia, and the immunoassay platform was launched in 2006. Our headquarters are located in Uppsala, Sweden, and we also have a sales office in the US, UK, Germany, and France. And we have a distributor network covering Asia Pacific, Middle East, Africa, and Eastern Europe. To exemplify how Gyros has transformed immunoassay performance and productivity, we here have an example of an assay conversion that was originally presented by Pfizer at the Gyrolab seminar. What we see here is a comparison for a generic PK assay between a Gyrolab assay and an ELISA assay. Some of the key things for the customer here was smaller sample volumes, going from over 100 microliters down to less than 10 microliters. 10-hour assay runtime with the ELISA was reduced to less than 1 hour with the Gyrolab. The dynamic range was extended both in the lower end of the curve and the upper end, so a broader range and more sensitive assay. The MRD was lower with the Gyrolab assay, and it was possible to do a 1 in 2 sample dilution prior to analysis. The system has built-in automation, which enables the possibilities to run overnight. Also, frequent interruptions from desk work. This means that once you have loaded the Gyrolab with your samples and reagents and CDs, then you can just press run and come back and analyze the data when it's convenient for you. So the sample processing is fully automated. These are the applications where the Gyrolab is typically used within bioprocess as well as preclinical and clinical trials. The presentations today will be covering applications where the regular bioassay CDs have been used. In addition to these, we also have a mixing CD, which is a CD that has a mixing chamber inside the CD that gives you the possibility to do sample pretreatment in an automated fashion inside the CD. This could, for example, be used for immunogenicity testing if you wanted to do an automated acid dissociation of your samples. The Gyrolab software is 21 CFR Part 11 compliant and can therefore be used from discovery all the way to post-market surveillance. The platform has been validated in both a GLP and GMP environment. Gyros immunoassay platform is based on a nanoliter scale flow-through technology. This is a picture of the instrument, and on the left side of the instrument is where you load your samples and reagents, and on the other side of the instrument, up to 5 CDs can be loaded. In the middle of the instrument, there is a liquid transfer arm that transfers samples and reagents from the microtiter plate to the CD. All assay steps, including incubation, washes, and detections, take place inside this fully automated system. The nanoliter scale workflow flow-through system combined format leads to small volumes of samples and reagents, and the automated assay workflow improves accuracy and precision. And these two combined shortens the total assay runtime. 1 CD can be completed in 1 hour, and up to 5 CDs can be processed in an unattended run. This slide shows you how we have implemented immunoassays on the Gyrolab CD. So the actual reactions take place inside the CD. Starting with a streptavidin-coated bead, you build your assay with a bioanalytic capture reagent and detect with a fluorophore label detection reagent. The streptavidin-coated beads are packed into a 15 nanoliter affinity capture column that is placed at the bottom of a microstructure. 1 microstructure equals 1 data point, and the other part of the microstructure above the capture column is where the sample volume definition takes place inside the CD. The CD surface is hydrophilic, and at certain key positions within the CD, there are hydrophobic barriers that stop the liquid flow. These are very important to get good accuracy and precision when working with nanoliter scale volumes. The microstructures are divided into different segments with 8 microstructures per segment and 12 to 14 segments per CD. After a run, a standard curve is generated, and we typically get 3 to 4 log dynamic range. In addition to this data, we also have a column profile generated for each data point, and all this data is generated in less than 1 hour. This is a short movie that shows you what happens inside a segment of the CD during an assay run. So first, the capture reagent is added to the common channel. Liquid goes into the CD by capillary action and stops at the hydrophobic barrier. Then when the CD is spun, we spin the liquid over the hydrophobic barrier down over the affinity capture column where the bioanalytic capture reagent binds to the streptavidin-coated beads. Then the samples are added to the individual inlets. The whole volume definition chamber is filled up with the sample and also the overflow channel on the side of the microstructure. Liquid goes in by capillary action, and the liquid flow stops at the hydrophobic barrier. First part of the spin defines the liquid, defines the sample volume inside the CD, and then the sample is spun down over the column where the capture reagent captures the analyte of interest as it flows over the column. In the next step, the detection reagent is added through the common channel. Liquid goes in by capillary action, stops at the hydrophobic barriers. Then the CD is spun, and the detection reagent flows over the column where the fluorophore label detection reagent binds to the analyte of interest. We now have a full sandwich assay which can be detected inside the instrument. In this particular example, we're just showing a capture, analyze, and detection step, but in a real assay, there are wash steps in between these different steps. In the previous example, a sandwich immunoassay format was shown, but it is of course possible to work with many other assay formats as well, and some examples are shown in this slide. Because of the flow-through format with very short contact time on the column, there is typically a reduction in matrix interference. In this nanoscale format, the distance between your capture reagents and analyte of interest is very short compared to, for example, in an ELISA well, which enables a much shorter contact on the column. The short contact time allows for the specific high-affinity reactions to take place, while there's less time for the non-specific bindings to happen. And therefore, matrix effects caused by non-specific bindings are typically reduced. Because of the high capacity of the beads in the CD, we typically get a broad dynamic range of 3 to 4 logs. In addition to the broad range, there are also different types of CDs available with varying sample volume, from 20 to 1,000 nanoliter. By adding a larger sample volume, a more sensitive assay can be developed. And on the other hand, if you add less samples to the CD, you can develop a less sensitive assay and thereby avoid having to do many dilutions up front if you're working with samples where the analyte concentration is very high. The combination of a broad dynamic range and different CD types allow us to work in the range from picogram per mil up to milligram per mil range. The instrument comes with a control and analysis software, and both these softwares are 21 CFR Part 11 compliant. In addition, there are different software modules available depending on application area. One example is the Gyrolab Viewer that gives you a picture of the fluorescent binding on each column in the CD. This is a very useful tool during assay development as well as troubleshooting, and I think we'll see an example of that in one of the presentations today. To support regulatory compliance, we also have other products. One is the Gyrolab LIMS interface that gives you the possibility to, for example, work with Watson LIMS and both export your work list from Watson and then import your data back into the LIMS system. We also have a Gyrolab functionality check kit that verifies the performance of the instrument in 20 minutes, and this can be used as part of your routine maintenance of the instrument. And there is also IQ/OQ validation and PQ guidance available. So to summarize, before I hand over to our first speaker, Gyrolab provides high-quality data, which reduces the sample volume requirements and thereby also the number of animals needed for a preclinical study. The automated assay workflow reduces analyst time and also speeds up time to results. And this is important when working with both preclinical and clinical assays. Now we will hear more about how the Gyrolab has been used in both a preclinical and clinical projects. But before I hand over to our first speaker, I just want to make you aware of the next webinar that Gyros is hosting. And that's a biomarker focus for this webinar, and it's on April 23rd. And it's 2014, April 23rd. And also, I just want to let you know that the recording from this webinar will be available online via Gyros or CHI website. So with that, I will hand over to Sufyan Makboul, who has a master's degree in drug discovery and development. He currently holds a position as associate scientist at Novimmune in Cambridge, UK, where he works in a GLP-certified bioanalytical lab with both animal and human studies. Sufyan has several years' experience using the Gyrolab for preclinical and clinical programs, as well as impurity testing. Today, he will share his experience using a universal approach to detect human IgG in animal matrix.
Hi, everyone. Thank you, Carolina. I'm Sufyan Makboul, and I'm part of the Clinical Pharmacology and DMPK group at Novimmune, Cambridge, UK. I'll be going through the detection of human IgG in preclinical matrices, a universal approach. So it's a PK assay that we've developed to support our preclinical studies. I would also like to thank Gyros for giving me the opportunity to talk at this webinar today. We have a long association with Gyrolab dating back to 2005, and Carolina, Anne, and all the other colleagues have been really helpful over the years. So we do value our relationship with Gyrolab. So my talk today would be the introduction to the Universal Assay that we've optimized, developed, and validated, and been supporting various projects across our pipeline at Novimmune. So we'll talk about the Gyrolab assay, present some data, and then show some comparison with MSD, which is another platform. So we'll do some comparison and draw some conclusions. So Novimmune is global biologics research and development arm of AstraZeneca, acquired in 2007. And we are committed to making a difference in science, in the lives of patients, and in the communities in which we operate. And MedImmune believe in spirit of innovation, heart of inspiration, and strength of integration. So clinical pharmacology and DMPK group is part of translational sciences, and we are a global group with colleagues in Cambridge, UK, Gatesburg, and California in USA. We acquired our GLP certification in 2012, and so it's just under 2 years. And so we support all our GLP tox and other associated studies. We develop and validate PK, PD, and immunogenicity assays to support preclinical and clinical trials of our large molecules. Predominantly use Gyrolab, and we have 2 validated equipment in our lab, and we have an MSD, so most of the work is divided between these 2 platforms. We don't really use much of ELISA now, and we have Vehicle for reagent characterization, and hopefully we'll be moving to ADA assays using the BA core. Ocean, another new technology for PK and ADA assays, and we use TechN for pre-dilutions. And we do assay development using animal and human matrices, and we work across the whole of our pipeline, which is across different therapy areas, so oncology, neuroscience, cardiovascular, metabolic diseases, infectious diseases, and neuroscience. So when we were supporting our colleagues in research, we felt we were spending too much time optimizing assays to support the early pharmacology studies and other preclinical studies, and we were looking for antibody pairs to be used as detection and capture reagents. So we felt that we needed something which would be more universal and which could detect the immunoglobulins in any animal matrix. So we don't have to search for these antibody pairs again and again. And we would then support our preclinical studies, and then we switched to more specific reagents, the NTAs, for our clinical studies. So we wanted something for the preclinical side to support the research colleagues. And we were thinking to harmonize across different sites, and then a method which we could roll out to other colleagues, so that would be multi-group and multi-area use. So we were ideally looking for some commercially available reagents, and that could be used on Gyrolab and MSD. So we have contingency plans in place, so we didn't want to rely on just one platform. So we wanted something which would work on at least 2 platforms. So the options we had for the Universal Assay were commercially available ELISA kits, and we worked closely with binding site in providing them assistance in our analytical support to develop their ELISA kit, which had really specific reagents which were cross-adsorbed to remove any cross-reactivity. So we supported them, and however we had problems where the dynamic range was a big issue. So it ranged from 2.5 to 810 nanograms per mil. Ideally, we would have 2.5 up to 50,000 on Gyrolab. So you can see we were losing 5-fold in the dynamic range. So this wouldn't work for us, and it was only a colorimetric ELISA. So as we know, Gyrolab uses fluorescence. So we wanted something which is more robust, and obviously the cost issue. So we thought we would take matters in our own hands and develop something in-house, which we would have more control over, reagents and day-to-day running of the assay. So commercial ELISA was a no, in-house was the way forward. So we had an old Universal Assay adopted from one of our sister sites in California, and basically they were using a sandwich ELISA with polyclonal capture and polyclonal detection, which they were using, and we thought, okay, we'll give it a go and see how this works. So we tried this on both Gyrolab and ELISA format. This wasn't run on MSD, so we didn't have good reagents to show that this would work nicely on MSD. So we went ahead with the Gyrolab and the ELISA format, but we soon realized we were facing problems. Robustness was bad. Recoveries of QCs were typically in the range of 30%. Ideally, we like to work around 20%. Reproducibility between operators were not meeting the criteria. Took time to develop, optimize, and qualify the assays. Hence, there was a delay, and then discrepancy between molecules. So we couldn't use the same assay to support different molecules across different therapy areas. So we used that for 2 years and thought it was time for change. So we then decided to have something in place which is more robust. So this new universal assay consists of a mAb antibody and a polyclonal detection. So we had the option for 5 commercially available mAbs were identified as human-specific, and then we tried the following MEDI molecules. So we had IgG1, IgG2, IgG4 isotypes, and so we initially tested them in PBS and 10% cyano serum. And what we saw was only one antibody recognized all MEDI products and was not cross-absorbed by cyano matrix. So this was the antibody 3. And as you can see here, these straight lines are the antibody curves in buffer, and the dotted lines are the antibody curves in 10% cyano. And as you can see, we've got nice linear curves. This wasn't optimized at this stage, so it was just a very quick look-see experiment. But we can clearly see that the signal isn't being absorbed out by the cyano matrix, and we've got nice linear curves. So we took antibody 3 with the antibody A, which was the MEDI product IgG2, and then we did a full curve using capture at standard 100 micrograms per mil, and the antibody 2 detection, which was 15 nanomolar, and we got nice linear curve, really good dynamic range, and then we saw really low backgrounds. So this was working, so we thought, okay, we would test this across different molecules throughout our pipeline. So as you can see, we've got oncology, neuroscience, RIA, and CVMD, and then we've got isotypes ranging from IgG1, and then we've got a fusion protein. So we've got different isotypes, tested different matrices, MRDs, ULOQ, LLOQ, all depending on what the requirements were. So we have no doubt that we can have really sensitive assays on Gyrolab, but it's about the practicality of the whole Gyrolab assay system. So we can go as sensitive as we want, but in these assays, these were the requirements, and these assays were deemed fit for purpose, and we met our precision and accuracy criteria. So from our point of view, we've been using this method now for a long time now, and it's very robust, it's consistent across analysts, across days. So it's working nicely for our preclinical studies. So that brings us to MSD, and basically we just wanted to see how these reagents would work on MSD. So we can split our workload on 2 platforms. So MSD is the electrochemiluminescence detection technology, uses sulfur tag labels which emit light upon electrochemical stimulation initiated at the electrode surface of multi-array microplates. So the amount of light emitted is directly proportional to the amount of analyte present within the sample, which is detected by the charge coupled device camera within the sector imager instrument. And so that's how the MSD works. So again, we had a sandwich ELISA format with bioanalytic capture, sulfur tag label detection, and we went ahead with this approach. We did some comparison using streptavidin and high-bound plates, which are provided by MSD. And as you can see, both curves on both plate types were exactly the same. We had really nice dynamic range, nice linear curves. These extra points on here are the QC samples on there. So we met our accuracy precision criteria. QC range was very similar to what we had on Gyrolab. Again, we know we can go, really sensitive on these platforms, but it's all about what the requirements are from the project. So no doubt about the sensitivity on these platforms. Then this table shows a direct comparison between Gyrolab and MSD. So the aim was to develop the PK assay using rat serum, and you can see the standard curve range pretty much identical. MRD, ULOQ, all the parameters were very close between the two platforms. So it's nice to see, that the reagent pair works nicely on both platforms. So if we look at the advantages and disadvantages, surely Gyrolab has the edge over MSD. When we're working in preclinical environment where the sample volume is a big issue, Gyrolab is a clear winner there. Also, the assay time is only 1 hour. Sample throughput, we've done up to 4 CDs in one day, which equates to 130 samples in a day. And then the really good thing is the Gyrolab Viewer, which is a really good troubleshooting tool embedded within the software. One slight downside could be the high maintenance of Gyrolab and the training required for the personnel. Carryover not such a big issue now, as Gyrolab have come up with pH 11 buffer, and my colleague Kabir Hussain presented last year at the Gyrolab conference where we did a head-to-head comparison between high salt wash and pH 11. So the problem we were seeing was high salt wash deposit within the reservoir. So we typically don't run any assays overnight using high salt wash because of the salt deposit. So by using pH 11 buffer now, we are much more comfortable using a wash to method, and we have now options to run our assays overnight. So it just works really nicely for us to have assays on Gyrolab. So to summarize, Gyrolab method eliminates the need for antibody pair screening. So this universal method that we have in place, which minimizes development optimization time, which is already less on Gyrolab, but using these reagents is even more quicker. So Antibody 3 has had a significant impact on the method, improved accuracy and precision and robustness. So the new Universal has the potential for cross-site, cross-group, and cross-therapy area harmonization. So Gyrolab provides robust assays with broad dynamic range, and associated parameters are always within, really tight data. Small sample volume allow lots of flexibility in the preclinical settings where sample is limited. By developing a universal assay on Gyrolab, our team can now focus on other tasks and then, provide data more quickly instead of, spending time looking for the right reagents. And lastly, the Gyrolab Viewer enhances troubleshooting options. So all in all, lots of benefits, and it allows us to work smartly in this time where projects are escalated very, very quickly. So Gyrolab allows us to meet our targets and always deliver on time. And that's all I have today. And I would like to acknowledge Joe and Tim, who've been really supportive to set up this assay within our group at Novimmune Cambridge, and other folks within our group, Carolina especially for her support from Gyros, and Chris Walker from MSD. And then I would like to conclude now. Thank you.
Thank you very much, Sufyan, for a very nice presentation. So we have several questions that have come in throughout your presentation. The first one is, I first have a question more on the technology in general that I will answer, and then we'll have more questions on your presentation. So it's about adding of reagent, the CD. So do the reagents need to be added manually between each spin, or are they all loaded simultaneously? So the reagents are added to the CD, either to the individual inlets or to the common channel. That's where you add your reagents. You fill up the whole CD or all the structures that are going to be processed, and then when you spin the CD, you will spin down all samples or all reagents at the same time over the column. So it's a parallel processing of all samples inside the CD. I hope that answers the question. And now we'll move over to some questions for you, Sufyan. So the first question is that I would like to know which type of CD was used and what kind of method. So I think for the method, it's in particular how many washing steps you were using, and also if you saw any carryover between CDs.
Yep. So the CD type was 200 nanoliter Bioanalysis, and wash steps. So we were using wash step 1 and 2. So we were using the CAD with wash 2 method. And last one was, carryover. So we typically in our group here evaluate carryover right from the beginning. So when we start our method development, we would evaluate carryover using the Gyrolab carryover test, and then use appropriate reagents. So we saw carryover with this assay, and then we've used high salt wash, and we've now switched to pH 11. So we did a comparison, and it was all very similar. So we've now moved to pH 11 wash buffer.
Yeah, okay, thank you. I think for the method, I just wanted to add some information in case we have some people that may not be as familiar with the Gyrolab technology. So in the method, you can choose to run with a more stringent needle wash, and this is to make sure that if you have a more sticky protein, that the needles are clean for the next sample transfer. In addition, we also have what's called a requisite buffers that are designed for dilution of samples and reagents to be used within the Gyrolab system. And this is to prevent any loss of reagents or analytes inside the needle. So it's a combination of a buffer for dilution and a buffer for washing of the needles that are used to minimize any carryover in the system. We also have a question about the concentration of the capture antibody, and if it always requires a concentration of 100 micrograms per mil. Is this something that you have looked into in your lab, Sufyan?
Yeah, we've gone with the Gyros recommendation, which says 100 micrograms per mil, and we always use the same concentration as we've done for all our assays.
Thank you. And I think in my experience, we typically go for 100 micrograms per mil, but there are assays where we may have used a lower concentration. But what's important is that it's to saturate the column with something that is biotinylated to avoid having unbound streptavidin on the column. We also have a question about preparation of your sample standards in QC, and if you do this using automation.
We haven't standardized a method as yet, but we are evaluating in using automation for the preparation, but that's still in progress.
Yeah, thank you. And we also have a question if you have inserted additional needle wash into the Gyrolab method to clean deeper and avoid carryover. And I think we kind of touched on this earlier as well.
Yes. No, we haven't inserted additional washes between the run. This was sufficient. Wash 2 was sufficient, and I can't remember using any additional step for any of the assays that I've worked on.
Yes. So you're using the standard method from Gyros with using two different wash stations.
Yes, that works nicely.
Yes. And we have another question. So how would you address the question of specificity? If this method were used for a human monoclonal drug entity, it would not be possible to use in the presence of the concomitant medications or biologics.
We don't have any contaminants within our PK for the PK assays, because all we're doing is the preclinical work supporting to GLP tox. And we do assess specificity during method development and then validation.
Yes, so and then you probably have other specificity for the reagents.
Yes.
And then we have a question: could the lower LOQ on the MSD versus Gyrolab be related to the longer incubation time or contact time?
Yes. So we've seen on MSD, as there's more incubation time, that does affect sensitivity, and Gyrolab being quicker result in more sensitive assays.
And I also saw that when you showed your data with the comparison, you were showing for, I think, rat matrix. Did you also compare the MRD or matrix interference for cyno and mouse between the two technologies?
Yes. So matrix effect was less on Gyrolab using cyno and the rodent matrix.
Okay. Thank you. I think we will move on and with Emily's presentation now, and then we will have time for more questions in the end of the webinar. So Emily Escoffier is a research assistant at Novimmune in Geneva. She got her Master of Biochemistry and Biotechnology from the National Institute of Applied Science in Lyon, France. Emily has worked with the Gyrolab for over 2 years and have experience from both PK and ADA assays from development to clinical sample testing. In addition, she has gained extensive experience from assay validation and assay transfer to a CRO on the Gyrolab. And this is the work that she will share with us today.
Hi everybody, thanks Carolina for this presentation. So as you explained, I will speak today about pharmacokinetic assays developed on the Gyrolab at Novimmune and its transfer and validation to a contract research organization. So to start with, I will first present briefly Novimmune, then give you the background of the project and enter into details on transfer and validation. And as Carolina said previously, I will say a word about the Viewer module on the Gyrolab evaluator and conclude on this presentation. So Novimmune is located at Geneva in Switzerland, and it's a drug discovery and development company focused on the creation of therapeutic monoclonal antibodies for the treatment of inflammatory diseases and immune-related disorders. 7 monoclonal antibodies have been generated, and we get a partnership with Genentech in 2010 for an anti-interleukin-17 monoclonal antibody. So for this project, we wanted to develop a pharmacokinetic assay for a first-in-class therapeutic monoclonal antibody targeting an inflammatory membrane receptor. We were looking for a technology with a low sensitivity, a wide dynamic range, but also allowing fast turnaround times, allowing treating many samples at the same time. So the development strategy was to generate tools in-house and to test them on 2 technologies, ELISA and Gyrolab. In terms of sensitivity, the ELISA and the Gyrolab were comparable, but in terms of dynamic range, the Gyrolab was better, so we decided to go for the Gyrolab technology. I will just briefly explain the technology. I know that Carolina explained it, but maybe for people who just joined the webinar could be useful. So the Gyrolab technology is based on CDs with the particularity to have microstructures, and each microstructure has a column coated with streptavidin beads, allowing a capture region bioaccumulating to bind to. So then you add your analyte, and it is detected by an Alexa through a labeled region. And the signal can be seen under the Gyrolab Viewer module. So I will discuss this part at the end of this presentation. So the samples are loaded in the microtiter plate and then transferred into CD thanks to needles. And thanks to the capillary and the centrifugal action, the samples are delivered into the colon, and then the signal is generated. So back on the project, we ended at the development with a bridging stepwise immunoassay consisting of the beads from the CD, anti-idiotype biostimulated mouse species, the drug, and the same anti-idiotype labeled with Alexa fluoro. So as the development was well completed, we decided to validate the assay. So it was done in 2 steps. First, the transfer to the CRO, and then the validation. Before answering into the data, I would like to answer this question: why transfer to a contract research organization? So as part of small and medium enterprises, Novimmune has limited internal resources, so it's sometimes very useful to have partnership with CRO. And for this study, we decided to go for a partnership with a European CRO located in UK because the study was European and we had previous experience with them. The requirements for to choose the CRO were the availability of technologies. Of course, we were looking for a CRO with the Gyrolab, also the expertise on this technology, and also on the assay, the cost of the study, of the transfer, and the capability to meet the timelines we are asking to. So to enter into a transfer, the goal of the transfer is to verify that you have the same data at your site and at the CRO site. So I will show you an example of inter-site variability assessment. Then you want to ensure that the assay is fit for purpose for validation. So this is done through the inter-assay accuracy precision, selectivity, dilutional linearity, and short-term stability assessment. So freeze-and-thaw cycles and room temperature. So for transfer, only few parameters are assessed because most of these points will be detailed during validation. The goal is just to ensure that everything is ready for validation. So here is an example of the inter-site variability assessment. So the concentration, drug concentration is the x-axis, and then the response in the y-axis, and the curves were very comparable. So we were happy to see that there were no inter-site variability. Then the transfer properly spoken happened, and we were happy with the different parameters assessed. So the transfer allowed us to set the low, medium, and high QCs, but also to determine the range of quantification, so from 4 nanograms per mil to 6,900. And as everything went smoothly, we decided to go for the validation. So as some of you certainly know, for clinical trial, it's required to have a validated assay to maximize the scientific integrity. So we wrote a protocol with the CRO, and all the data were reported in the validation report. And this validation report is used for clinical submission. So now I will detail the inter- and intra-assay accuracy and precision, the selectivity, dilutional linearity and hook effect, and then the short and long-term stability. But before entering into data, I would like to briefly remind these 2 common terms because they are very used in the field of validation. So it is accuracy and precision. The accuracy measures how far you are from your targeted value with the determined value you get. And the precision, assessed through the coefficient of variation, is measuring the scattering of your data. So as a picture says 1,000 words, here are schematic of what you can obtain. So the most expected case is the first one with precise and accurate data. But you can have very precise and inaccurate, so far from the target you are expecting to reach. And you can have accurate but very scattered data, and of course you can have the two problems, but for validation, of course, you are expecting to have the assay as precise and as accurate as possible. So for this validation, the criteria were accuracy less than or equal to plus or minus 20%, and coefficient variation less or equal to 20%. And these criteria are based on the EMA guideline, the last one. So to start with validation data, here is the inter-assay accuracy and precision for the calibration curve. So 10 points are represented with 2 anchor points with no criteria, and the LLOQ and ULOQ points. This curve was assessed in 6 different runs. If we focus on precision and accuracy, you can see that every data, all of the data are within acceptance criteria. Concerning quality control, so first the intra-assay, 5 labels were assessed in the same run in 9 replicates, so at the lower limit of quantification, low, medium, high QC, and upper limit of quantification. Again, when focusing on precision and accuracy from this for this run, we can see that the values were within acceptance criteria. Now the inter-assay, so again the 5 levels were assessed, but this time in 3 sets, and it's 6 different runs. And again, in focusing in the precision and accuracy, we can see that all the data were within acceptance criteria. Another parameter important to assess is the selectivity. So it measures the inter-donor variability, and it's very important to know before entering into clinical trials if some matrices could lead to an increase or decrease in the signal just because of the matrix, not because of the assay. So first, in normal serum, 3 levels were assessed: blank, low, and high QC. The blank level returned values below the limit of quantification, so that was expected. And for the low and high QC levels, 20 donors were assessed, and you can see the accuracy with the green boxes and the precision with the line graph. And the limits are stand by the purple lines. So as you can see, for the low QC, 100% of the donors were within the criteria, except 1 for the high QC, but this was completely fine for the validation. So to continue on selectivity, it's also highly recommended in the last guideline to assess lipemic and hemolyze matrices. So this is done by adding triglycerides or whole blood into matrices. And this was done at the low QC level. And again, same kind of graph with lipemic matrices here and hemolyze. And we're happy to see that no problem with these specific matrices. The dilutional linearity and hook effect. Basically, you want to be sure that if there are samples out of range that you need to dilute, you want to be sure that the dilution does not affect the accuracy and the precision of the value. So the dilutional linearity was performed by diluting using different factors and through the range of quantification. And we started to be not as precise as before at this factor of dilution, 5,000. So we conclude on good linearity up to 2,560 in addition to our MRD. And this is far enough because as the ULOQ is already high and we expect to have samples that we won't reach this value if you multiply this factor with the ULOQ. Concerning the hook effect, samples at 1 milligram per mil were assessed and the return value higher than the ULOQ value, so we were happy to see that no hook effect was observed. So to go for the stability assessment now, at short term first, so at room temperature, 8 hours and 24 hours were assessed, and this is for example, you leave your sample a day on the bench or a night, and you see just the next day on the bench. So you want to be sure that your sample is still used, you can still use it. So as you can see, for low and high QC, 4 sets were assessed, and everything was again within acceptance criteria. So we concluded on 24 hours stability at room temperature. Then for 3 cycles, we were looking at how many cycles we could perform with the samples. So we conclude that 5 cycles were fine, and this will be used for sample testing. And concerning long-term stability, we assessed the 6-month stability for long-term stability, and it has passed at 20% and 80%, 20 degrees, minus 20 degrees and 80 degrees. So to conclude on this data, the assay was robust, and it continues to be robust in clinical sample testing. But as you certainly know, as part of the process, you don't always get these perfect data, and some assays could be more challenging to validate. So it would be my message for today: okay, you can have good assay, but could also have more challenging assay. To change theme now, I would like to be more technical and to explain you what you can do with the Gyrolab Viewer module. So as Carolina explained you, it provides an access to the binding profile for each data point directly from within the Gyrolab evaluator. So as you can see here, the intensity in the y-axis, this is the column, and this is the beginning of the column, and the flow go from left to right. And you can click for auto color, and you have this nice coloring of the peak. And in white, it's the integration area that is used by the software to calculate the concentration. As Carolina said, again, it's powerful to visualize affinity binding, of course, but also to identify the optimal pairs, so best capture and detection antibodies. Also useful to troubleshoot and also to investigate outliers. And this part, we use it during validation. So as some of you may have noticed, there were 2 stars here for these 2 QCs, and it's a result of 1 replicate. So what happened is that it was clear outliers for one of the 2 duplicates, and we investigated the value under the signal under the Gyrolab Viewer, and it was atypical profile. So we had a scientific justification to remove the point and to have the run valid, because otherwise the run should have been repeated. So what is atypical profile? Maybe before, what is normal profile? So it's a well-distributed peak with intensity less than 1, so not saturated. And this is for a medium level, I would say, but you have also normal peak for lower levels. So one type of atypical profile is called clogged column. A key characteristic is that all the signal is at the beginning of the column, and this is caused to a particulate matter getting caught at the beginning of the column and avoiding the flow to go through the column, the flow leakage, sorry. So to prevent from that, Gyros company recommend to filter buffer, to use Rexip buffer, and to centrifuge samples if that is possible. Another type of atypical profile, very easy to recognize, is what we call fluorescent spike. So it goes to the Alexa fluoro region, or any particulates that could generate fluorescence, and you can see that it's at a specific signal. So to avoid this problem, try to centrifugate the detection and to pipette only the parts without aggregates. And the last one I would like to present you is what we call negative value. So maybe for people who are not very familiar with the Gyrolab, there are two readings during Gyrolab method. First at the background level, and then the signal you have generated thanks to your assay. And if there is contamination at the background level, when it's subtracted to the second signal, you can end up with a negative value. So you can see that it's a kind of hole, and meaning that you have no signal, it's negative value. And to finish on that, you can see that you can have many other atypical profiles, and I'm proud to say that all of these atypical profiles are from Novimmune during validation. But you can see that it's artistic, but you won't learn so much by looking at each data. So my advice would be look only if you have a need or interest into looking at every data point. So for us, it was a good justification to allow exclusion of outliers. And I would say that it's not always easy to find the solution to overcome the frequency of these profiles, but it's good to follow all the recommendations to try to decrease this frequency. So to conclude on this experience, this presentation, we developed fit-for-purpose PK assay, and we transferred it to the CRO. The transfer was smooth, and we got robust data, and we successfully validated the assay on the Gyrolab at the CRO. And we were happy to have the Gyrolab Viewer to investigate outliers and remove the atypical profiles we obtained from the final data. So I would like to acknowledge in front of this beautiful mountain, which is a Mondo, my head, Sabrina Lori, Maureen Dehan, Walter Felin, and also my team. And of course, thanks for Gyros company to giving me the opportunity to present on this subject. And also, thank you for your attention.
Thank you very much, Emily, for a very nice and informative presentation. We have several questions coming in, so let's start with those right away. One question that came in was whether there are any examples of validated PK assays on the Gyros with LLOQ lower than 5 nanograms per mil. And I think in your example, your LLOQ was lower than 5 nanograms per mil.
Yes, that's true. But I think it's really linked to the anti-adiotype, which was the best obtained in-house, which was selected, and we didn't. I think it's one of the best anti-adiotype we got, and the best lower limit of quantification for in-house assay.
I agree with you. I think the LLOQ is very much going to depend on the affinity and quality of your reagents, as well as, of course, the MRD that you can run the assay at. There was also a question earlier on the binding capacity of the column, and the binding capacity of the capture column in a bioassay CD exceeds 100 femtomolar. So that's the answer to that question. Then we have some questions that are related to selectivity and specificity. So how selective is Gyrolab assays for endogenous interferences and also interference from drug-related metabolites?
We have not experienced these problems.
Yes. Sofiane, do you have any experience that you would?
I think from our perspective, the short incubation helps to overcome these problems.
Yeah.
And the metabolite and other things are more of a small molecule sort of problems, analytical problems. So don't think it directly applies to large molecules.
Okay, thank you. Another question that's also related to selectivity and specificity is if the lipemic and hemolyte tests were done in the ELISA as a comparison to your Gyrolab data.
You mean for the selectivity?
Yes.
If we have compared in ELISA?
Yes.
We have not done, we just had compared in terms of sensitivity and quickly accuracy and precision, but we don't used to go into details so different matrices. I think what quick selectivity may be on 5 or 6 donors, but we haven't performed complete selectivity on the 2 technologies.
And in your previous experience with ELISA?
So the question is it better or comparable, is it?
Yes.
I would say it's really depending on the assay, on the format, not really the technology.
Okay, and then we also have a question if the selectivity for the hemolyte or lipemic samples were only tested for one QC level or for several QC levels?
For the low QC only.
For the low QC.
I can, if you can keep this question, I can check and come back to the person who has this question.
Yes, perfect, thank you. And have you tested diluted sample stability? So, for example, test to store your samples at MRD at 2 to 8 degrees for multiple days prior to analysis.
No, each time the samples of the QC are frozen in neat serum and then diluted to the minimum required dilution before being tested. So we have not tested the stability at the MRD.
Next question is related to transfer from an existing ELISA to the Gyrolab platform, and how easily can an existing ELISA method be transferred to the Gyrolab and vice versa?
We have done what we often do is to first develop on ELISA because honestly it's cheaper, but we have never experienced problem to transfer an ELISA assay to a Gyrolab assay. It's just about labeling, I would say, for the alexafluoral region, so detection. But I would say it's more get used to with the technology, but in terms of assay, to transfer from ELISA to Gyrolab, I would say it's easy.
And Sofiane, would you like to comment on this and your experience?
We don't typically transfer anything from ELISA. We just start straight on Gyrolab. So I think that, as Emily said, the labeling could be a problem. Especially we've seen that when we transfer our assays to CROs. I don't know whether Emily could answer. Did they provide their own labeled reagents, or did the CRO label their own reagents?
What we used to do is to have all the reagents ready in-house and then send the critical reagents, so the capture, the detection, and the drug to the CRO, so that they have we know what we have sent and we are sure of about the labeling and everything. So, and if we need to compare on site, I mean, to see if at Novimmune there is a difference with the CRO, we can better troubleshoot the data and to understand if it's coming from the labeling.
Yeah, and while we're speaking of working with a CRO, we have more questions on this topic. So at the CRO, did they do any of the preparations for the dilutions using automation?
Sorry, I'm not sure I understood the question.
So at the CRO, were any of the preparation steps for the dilutions of your samples done using automation?
Honestly, I'm not aware. I don't think so. I think it's manual pipetting, especially because not this assay, but another one is quite sensitive to small variations. So I would say that for small volumes, maybe better to have manual experience. I'm not, but I think the one we transferred this assay is working with manual pipetting.
So manual sample preparation and then...
Yeah, exactly.
How many analysts were able to run the method for this validation work? So when you validated the assay, how many analysts ran the assay?
I think it was 4. A special training was delivered to be sure that enough people were able to use the technology, and yeah, I know that the CRO put effort to have well-trained analysts on this technology.
How would you compare the ease of transfer of a Gyrolab assay to a CRO with other technologies in your experience?
I would say that as it's automated, it makes I would say, the one instrument is very similar to another, so you don't have problem in terms of signal. Should be very close to one another, and you have the same procedure performed by the instrument. I would say that it's maybe small volume handling that could be challenging, and also if you work with sticky molecules, could be complicated to remove carryover sometimes, because at CRO, we say CRO are working on different projects, so if you have a sticky molecule, it could affect other assays, so it could be complicated. But for this assay, it worked very well, and the Gyros was very good technology to work with.
Yes, and do you feel that the buffers that have been developed to clean the needles and for dilution of the samples have helped you solve your carryover problem?
Yes.
Yeah. Another question regarding outsourcing and transfer to a CRO. If the samples are only going to be analyzed at the CRO and not at your company, would you still need to do the assay comparison between the two sites?
So do you mean testing the samples twice?
So no, I think today you're doing the sample analysis both at Novimmune and at the CRO. But if you were only going to do the sample testing at the CRO, would you still develop and validate your assay internally and do a comparison between the two sites?
So you are speaking about another project. For this project, it's only the samples are testing only at the CRO. But for other programs, it's true we are doing a validation in-house and comparing with other sites. So it's really depending on the program.
Okay. Yeah. So it depends on the program if you do a comparison between the two sites. But in the case when it was only going to be sample analysis at the CRO, would you be fine with not doing the comparison between the two sites?
Yeah, we won't do the comparison if the sample testing is only at the CRO, or I mean, if the clinical sample design or the study design is not needed to have a testing in-house, so we won't validate in-house. We will directly transfer, validate, and the sample testing will be done at the CRO site.
Okay, thank you. Sylviane, is that anything that you want to comment on in your experience transferring an assay to a CRO?
We've seen as recent as last year CROs struggling to label, Alexa label in particular, when we were trying to transfer or asking them to actually perform the validation and assay development, but there seems to be a problem with labeling, and we've seen that recently again, that they were struggling to label, Alexa label reagents.
And how did you solve that? So are you labeling, or were you able to solve this?
So we had our reagent labeled in-house, and then it was transferred to CRO, and then they labeled the reagents, and they were compared against each other. But it seems to be the case that they assume things and not necessarily need to confirm small details. So, when Gyros recommend, the labeling, the molar ratio to be between 3 and 7, they've been using reagents that didn't fall within the range. So, seems to be, ignoring small details.
Yeah, so I actually have a comment from one of the attendees that they also think that they agree with you, Sofiane, that large-scale labeling is better done in-house or outsourced to RAD, was a comment here.
And it seems like they don't follow SOPs, and so we have to send our SOPs because we strictly follow guidelines given by Gyros, both for bioaccumulation and Alexa labeling. So I think they need to pay more attention to small details and somehow follow an SOP, so it's all recorded, and it's consistent across analysts. Then they're all trained to the same SOP.
Yes. Then we have some questions about the Gyrolab Viewer software. So is it necessary to check the profiles of each individual data point as a QC step, and does the instrument software recognize the atypical profile? And I can comment on this one, that the software does not recognize all atypical profiles. Some of them the software will highlight, and others the software may not highlight. And I think, Emily, you mentioned in your presentation that you typically check the viewer profile when you see an outlier in the data, and then you use the Gyrolab Viewer to justify exclusion of that data point. Is that correct?
Yeah, it's fully correct. And I would add that, of course, as you are in the frame of validation, you can't say, I will remove this value because I want it. You have to write when you will remove an outlier, so this should be set in the protocol. And yeah, you have to, I would suggest to include the profile, the view image in the report, so that you are sure that the authorities will be fine with removing a replicate from the data set.
Yes. And also, if you deactivate or remove one of the replicates in a duplicate of your samples by looking at the viewer picture, and then you're only left with one sample replicate, would you repeat this sample, or would you be okay with just using one replicate?
It's really depending on the assessment. So for the selectivity, as you have seen, 4 sets were assessed, so it was fine to remove one replicate because there were different sets assessed at the same time. I would say in an inter-assay where you have 3 sets should be fine too. I think it's depending on what you are assessing. During sample testing, I will definitely rerun the assay because I want to be sure of the data I will deliver. It's more about what you have in your run and about the statistics which are behind for the analysis, I would say.
Yes. And would you treat your standards and QCs in any different way than a sample here, or you would handle the standards and QCs in the same way?
So as part of the guidelines, for a standard, you need to have the wall point excluded. So anyway, even if you see an outlier, you will remove the wall point, so the 2 replicates. For QCs, it's different. I would say, if you see it's a clear outlier, I would say a signal 3 times what you are expecting. I think it's enough to say, okay, it's an outlier. I have the profile. You can see it's atypical, so I would remove it, just the replicate, and really explain that it's a result of 1 replicate and not a wall point. So you have a mean of just 1 point. So that's why I said it's important to know the the important, how it affects the statistics. So after it's more the statistical, you need to say, okay, I have enough data to have, confident, be confident on data with all the data you generated, even if you remove one of the data or many of the data.
Yes. So thanks a lot to both of you for sharing how you use the Gyrolab Viewer and how it could potentially be used. I think that these discussions will also continue at the bioanalytical workshop that will be in combination with the NBC meeting in May in San Diego. I think it's a very interesting topic. So in interest of time, I think we'll have maybe time for 2 more questions. Someone has heard that the temperature inside the Gyrolab may fluctuate during the assay. Do you have any experience from doing multi-CD runs and that it could potentially be a problem to run at room temperature?
We used to run at the maximum 2 CDs a run. I would say that we have not used so much the overnight run, so with 5 CDs and using all the capacity of the Gyrolab, so I don't have data on that, but I know that there are companies doing it and it's functioning, but I can't say anything about the overnight or running 5 CDs because we just run at a maximum 2 CDs a run.
Yes, and I think this is something that also would be analyze dependent, so it's something that one should probably look at when validating a Gyrolab assay. Some assays would probably be perfectly fine to do a 5 CD run in room temperature, while others you may only be able to do a 3 CD run, for example. So it's analyze dependent.
Just want to add about the PD assays, they might be more susceptible to, rise in temperature. So that's one thing to look out for.
Yes, thank you for adding that.
Okay.
So one question is also regarding the data output of the Gyrolab, if it's similar to MSD outputs, and in any issues trying to use a 4 or 5 parameter curve fit on the point, and is there a need for weighting or other curve fitting options? So do you want to comment on that, Sufyan or Emily?
We typically use the normal settings for the software, the Gyrolab software, and on some occasions we then might change the setting to get better curve fittings. We wait on response when analyzing samples.
Yeah. Emily, any comments from you?
I've just worked a little bit with the MSD. I just know that it's more complicated to make all the analysis you would like to on the MSD. It's on the Gyrolab you can directly weight and analyze with 5 parameters. I know it's only 4 parameters on the MSD, as Sufyan said, so maybe it's more flexibility on the Gyrolab for this part for data analysis.
And also one more question regarding the automation topic. So someone is interested to hear from other Gyros users if they have an interface to help create a script from the EvoWare using Watson worklist that contains the sample dilution, which would be compatible with the Gyros plate map. Is this something any of you have any experience from?
No, for me, no.
No, not for EvoWare. I think with Hamilton's you can use.
Yeah, and I think it's also a question to the audience. If anyone has any experience that they would like to share, that would be great. I know that we have one customer that has a publication out there regarding integration with a liquid handler. So I can see that we're running out of time, so I just want to take the opportunity to thank everyone for listening and for all the great questions, and of course a special thanks to our speakers for very good presentations, sharing their Gyrolab data. And again, the recording will be available on Gyros and CHI website. And I also want to remind you about the next webinar, which is on April 23rd, 2014. It should be 2014. All right, thanks everyone.